right, as we finish up A and P, we're going to go with the last unit in the last organ system, the reproductive system, which is really two systems because you have male and female reproductive system. But it's super interesting that whether it's male or female, embryonically, all of the organs start off in the same place as the same kind of embryonic tissue. And then, under the influence of the varying hormones, the testosterone or the estrogen produced by that embryo, it develops into the different systems. So there's counterpart organs in male and female. So we'll look at that. We'll see it especially next time when we start the reproductive system, and for females, and we'll compare some of those organs to what you're going to learn today with males. Okay, but with the reproductive systems, uh, there are the special organs that actually produce the sex cells, and those are called the primary reproductive organs. We'll see those. So in males, that would be the testes, and females, that would be the ovaries, and they produce the sex cells. We're going to see when we do female reproductive system that the sex cells, a lot of people just refer to them as eggs, but we're also going to see this term. That's not an oocyte, it's an oocyte. That's how you say that. Anyway, we'll talk about oocytes next time. But when sex cells are formed, they are very different from other cells of the body. Okay, if you were in a genetics class and we were talking about this in a lot more genetic detail, most of the cells in the body are called somatic cells, like liver cells, lung cells, muscle cells, all the other cells. But the sex cells are the gametes, or sex cells. And what makes them different is the number of chromosomes. As a human, our chromosome number is 46. You ever heard that number? Okay. But our chromosomes are in pairs. So you may have heard the number 23. Okay, we're actually in your somatic cells, all the cells except the sex cells. You have 23 pairs of chromosomes. And scientists have actually numbered the individual chromosomes. And what makes it a particular chromosome is what genes are located on it. So you have chromosome number 1, number 2, number 3, number 4, all the way up to the 23rd chromosome. And then you have a counterpart to each of those. So you have 2 number 1s, 2 number 2s, 2 number 3s, and so forth. As a human, when you were first formed as a zygote, you got one of your number 1s from the egg that formed you, from your mother, and the other number 1 from your father, from the sperm that formed you. Okay? So that's why they're in pairs. All right, and so the sex cells have to have only one member of each pair, one number 1, one number 2, one number 3. And so when they are formed in the gonads, or the ovaries or testes, it's not just through mitosis, it's through another kind of cell division called meiosis. Meiosis. All right, and so we're going to dwell on that a little bit early on as we look at the formation of egg and sperm in each of the systems, male and female, and we're going to see that formation. It includes some mitosis, but the bulk of it is through meiotic cell division. Okay, and the whole purpose of meiosis is to form cells that have one of each pair, one member of each pair. So in other words, the sex cells only have a total of 23, so that when they unite with the counterpart sex cell to form a zygote, there's a total of 46. Does that make sense? Okay. All right, and we're going to see that meiosis includes 2 divisions. The first one, meiosis 1, you don't have to write this down, but the stages in it are anaphase 1, metaphase 1. Excuse me, I said that wrong. Prophase 1, metaphase 1, anaphase 1, telophase 1. And so that results in 2 cells. And then as they go through meiosis 2, it's prophase 2, metaphase 2, anaphase 2, telophase 2. So I've got a video that's going to show you that. I am not going to test you on the individual stages within meiosis, but I want you to see it through this video, which is not on the next slide. It's a couple of slides after this one, But let me give you a quick little overview on this slide. Let's say this is a cell. This is a normal somatic cell in an ovary or a testis that is going to ultimately divide to become the sex cells. Okay, and so when it goes through meiosis, each of its chromosomes is going to do the same thing that it does in preparation for going through mitosis. In that chromosome, takes you back from AMP to AMP1. That chromosome, that long skinny chromosome, has to replicate, and where the chromatids are connected, that's a centromere. Remember that? Okay, so it's got to be replicated before it starts dividing. Okay, and then so let's say it's done that, and then the centriole pairs replicate. So all that kind of preparatory event's going to occur, and then in the first meiotic division, which you'll see on the video, is prophase 1, metaphase 1, anaphase 1, telophase 1. During that first division, in the metaphase part of it, remember in metaphase they line up in the middle? Well, instead of just lining up in the middle, they line up still as pairs. So here's the 2 number 1s, here's the 2 number 2s, and just imagine this being 23 pairs and not just 2 pairs. Okay? So they line up as pairs, and then in the anaphase 1, the pairs separate. And so that the resulting cells, do you see how they have one member of each pair? Okay. And then now what's wrong with that? That would be if we, made this 46 and 23 instead of 4 and 2. It's still, that has, the 23 chromosomes, but they're still in a replicated state. So that's why you have to go through the meiosis part 2, which looks just like mitosis, because the chromosomes now are going to line up single file, but now their chromatids separate from each other. Okay, you just have half the number of chromosomes. So you actually end up with not just 2 cells, but 4 cells. But now each new cell has the correct number of non-replicated chromosomes. Okay. Another thing that can happen, if you go way back to the first meiotic division to this one, when they're paired up, I think I've got that on this slide. Yeah. Let's say here's a pair. And so this is this long coiled up chromosome. It coils up during prophase. All right. Well, it doesn't just pair up in that first meiotic division. Look at what happens here. Some of the chromatids from the opposite members of the pair cross over and break apart, so that now we've got 4 different combinations of genes. There's one combination, there's one, there's one, and there's one. So you got a variety of genes. And so what I mean by that, like different combinations of genes, let's go back to the left over here, and let's pretend that at the tip of each of these chromosomes is a gene for your hairline. Either you have a straight hairline or you have a widow's peak hairline. I'm picking out some traits that they just come in 2 varieties. Okay. So you got a hairline gene at the top, but let's say on this one it's straight. If one's straight, the next is straight of these 2 because it was replicated from 1 chromosome, right? But then on this one you got a widow's peak gene. Okay, it's still hairline, and so that's what makes these homologs or pairs of each other. Okay, but see how they're different? Okay, so this particular person got a straight hairline gene from, say, its mother, and a widow's peak from the father of that person. Okay, all right, but on the other end, let's say this has to do with whether, what would be another? Your earlobe. You either have an attached or an unattached earlobe, nowhere in between. Do y'all know what that means? If you've had a genetics class, you know this. Let's say here's a person. This would be an attached earlobe. Now, you're not going to have both kinds in one person. I'm just going to draw it on one person. This would be unattached, where actually lobe hangs down a little bit. Whether it hangs down a little bit or a lot, it's still unattached. Okay, so let's say here's attached on this one, and this is unattached on that one. Okay? Still an earlobe gene. All right, when crossing over occurs, do you see now, after all this separates, you're going to have like 4 chromatids going to the 4 final resulting cells at the end of meiosis. And do you see how the first one's going to have a straight hairline gene combined with an attached earlobe gene. The second one's going to have a straight hairline gene combined with an unattached earlobe gene. And the third one's going to have a widow's peak combined with attached. And the fourth one has a widow's peak combined with unattached. So another beauty of meiosis is not that you just end up with cells that have half the number of chromosomes as the original cell, but they also have a different gene combination than the original cell, and they all have different gene combinations in each other. And that gives variety. That's why none of us all look alike. That's why we have so many unique characteristics, because these chromosomes can have hundreds of genes on them, and the crossing over can occur with all 23 pairs in several places, not just one place like this. So it's almost unlimited, the variety of gene combinations that we have. And that's the whole purpose of sexual reproduction. Whether you are a human being or a pine tree or a bacterium, the sexual reproduction allows for variety in the species to withstand harsh environmental conditions. We don't see that with humans so much because we can control our environment a little bit, but other organisms cannot. So anyway, it's called survival of the fittest. So you've heard about that. So let's look at this video, and this shows the picture I just tried to do a lot better, how it shows different combinations ending up in the gametes. And I've spent a lot of time on this, not because I'm going to test you on this crossing over or any of that, but you just need to see it, because I know a lot of you come into this course without ever having a genetics class, and it is very interesting, and it's important to see the variety that can result. Okay, so let's look at this video. It's way more detailed than I would ever test you on, but I want you to see the stages.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis I, halves the number of chromosomes, and in the second division, meiosis II, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase I, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just as easily have ended on top.
Okay, what that's all about is, let's pretend like the purple one initially came from the egg that formed this person, and the green one came from the sperm. So the purple one is the maternal member of the pair. The green one is the paternal member. And so do you see how they don't always line up perfectly? You could have, anyway. That's Mendel's law of independent assortment if you were in a genetics course.
Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, two cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have two cells, each with two chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only one of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber. And in telophase 2, along with cytokinesis, we see the formation of four genetically different haploid cells.
Okay. This next video, just trust me, it shows exactly the same thing. Okay, so we're going to bypass it. I need to remove it from the notes. Okay, so now that you understand a little bit about how the gametes, which is a fancy name for the sex cell, how they're formed, we're going to now look at, today we'll focus just on the male reproductive system. But you might wonder initially, genetically, what causes someone to be male or female. It has to do with that 23rd pair of chromosomes in that little zygote that's formed. The 23rd pair, if they are identical all the way down, if it's identical, meaning this may not have the same exact variety of a gene, but the same type of gene, like eye color or hair color, whatever, but maybe different varieties. But if they're all the same, those are X chromosomes, okay? And if you have two X chromosomes, you are a female. But there is another chromosome that is part of the 23rd pair in half the population. It's called a Y chromosome. And it has some gene traits similar to the X, and so it pairs up with it, but then it has other ones that are not. But it does act as a pair, and so if it's an XY for that 23rd pair, it's male. So if the 23rd pair is made up of two X chromosomes, it's female. If it's made up of an X and a Y chromosome, it's male. Okay, and so I'm getting way ahead and not even into this class, but just say if it's a female, XX, when she starts making eggs later on in her life, each egg, after it goes through all this meiosis, is going to have one X chromosome because she had two to begin with, right? Okay. But if it's a male going through all this meiosis to form sperm cells, because that male's 23rd pair was X and Y, do you see half of the sperm get an X and the other half get a Y? Okay, so there are X sperms and there are Y sperms, and whichever one is used in fertilization determines the sex of the new baby, okay, because all the eggs are X. Okay, interesting. So the moral of that is the female has nothing to do, the mother has nothing to do with the sex of the child. So Henry VIII is the one who should have been beheaded instead of all of his wives who wouldn't give him a son. Yes. Go back in history. Yes. All right, so let's look down at the male reproductive system. The primary organs or gonads, if I use the word gonad, G-O-N-A-D, that means the primary organs. Those are the testes because they're the ones that produce the sperm cells. All the other organs we'll look at are the accessory organs. Okay, so just to give you an overview of the male reproductive system. This is, of course, a sagittal section, and the testes are located in the scrotum, and there's two of those. Um, Probably, well, for sure. We'll come back to this picture several times, but just to get your bearings on this, here is the digestive tract, and then here's urinary. So remember, there's the bladder, and then, and we did this last time, goes through the urethra and out the penis, but the reproductive system, here it is. And see how it also empties into the urethra at that point. And so that's the reason that the urethra acts as a urinary and also a reproductive organ. It's interesting that the testes are located in the scrotum, which is outside the body cavity. The reason for that is because sperm cells cannot be formed at body temperature. They're formed at a couple of degrees below body temperature. Do you remember enzymes work at specific pHs? They also work best at specific temperatures, and the enzymes involved in sperm formation work at a lower temperature than body temperature. However, testes don't begin their existence in the scrotum because in the early embryo there is no scrotum. Okay, the testes actually begin their existence up in the pelvic cavity. This is what you look at this. See right there, and it's in an area where in the early embryo, it's just called gonadal tissue. And under the influence of testosterone produced by that little male embryo that has the Y chromosome, then it develops into testicular tissue. And then late during gestation, gestation being the 9 months of pregnancy, late during gestation, of the last couple of months, it starts to descend. So now you can see it later on, it's descending. And then by the time that baby's born, it's descended all the way down into the scrotum. So sometimes if there's a very premature baby boy, he may have undescended testes. Okay, that condition is called cryptorchidism. That's a weird name, cryptorchidism, undescended testes. But if a child is born with that condition, usually I think it's a wait and see approach because they probably will go on and descend on their own. And if they don't, they'll be surgically descended because not only would he not be able to produce sperm later on once he hit puberty, but also testicular tissue at normal body temperature is much more prone to infections, cancers, so there is a fibromuscular cord. There you see it right there. It's called, it has a funny name. There's a lot of weird names in this here. But it's called the gubernaculum. And it's a fibro, which means fibrous connective tissue, but also has some muscle in it. Fibromuscular cord. And that shortens and literally pulls the testes down into the scrotum. Okay? And it goes down through a weakened area in the lower pelvic wall called the inguinal canal. If a man has an inguinal hernia, women can have it too, but it's much more common in men. That's when the intestines loop down through that already weakened area. And but anyway, but ultimately, after the testes have completely descended, the tube that later on in life the sperm will swim through, it's called the vas deferens or the ductus deferens. Don't worry about writing it down. We'll see it later in the notes. But anyway, that tube, along with blood vessels and nerve fibers, are enclosed in a fibrous enclosure, and that whole thing is called the spermatic cord. Okay, everything's enclosed in, right? All right. Maybe you should know the word, the strange word, cryptorchidism. I think that's how it's spelled. Cryptorchidism. That is the condition of undescended testes. Okay. Yeah, it is in the book. Yeah. I just think I forgot to put that in your notes. I'm sorry. Cryptorchidism. Undescended testes. Okay. Okay. So now if we look at the mature testes, you stay on this page. Let's look at a diagram. Here you see a sectioned view over on the left. And the most noticeable thing about it, it's got these compartments in it. And in these compartments are curled up tubes. And this is not drawn to scale. Those tubes are really a whole lot more numerous and smaller. But they're called seminiferous tubules. You'll see that term on the slide just prior to this one. Okay, and that's where sperm cells are produced, is inside these seminiferous tubules. What you're seeing right here is a cross-section of one seminiferous tubule. Do you see those little sperm cells in the lumen, the opening of it, the lumen? All right, and so all of these cells along here, these are cells in varying stages of meiosis. So those are the cells that are producing the sperm cells, okay? And that area right here that produces the sperm cells is called the germinal, that's an E, germinal epithelium. Okay? Germinal epithelium. And then here's the real thing. You can see a seminiferous tubule. All right, so obviously the testes produce eggs, but no, they don't. The testes produce sperm. Let me stop for a minute. My battery's going out on this mic, and I'll put a new battery in it. So I'm just going to pause. Just hang on. You'll just hang on a few minutes. I'll come back. Okay, I'm back. I have a new battery now. Thank you for telling me, Whitney. Um, where was I? I forgot. Testes produce sperm. Yeah, testes produce sperm, but they also produce hormones like testosterone, right? The sperm are produced in the germinal epithelium, but the hormones are produced by these cells that are not inside the seminiferous tubules. They're in between the seminiferous tubules, and they're called interstitial cells or cells of Leydig. Interstitial cells or cells of Leydig. So here you see some. Here you see some right there. They're in between the seminiferous tubules. Okay? All right. If I ask you on a test, what is the name of sperm cell formation? Your answer is spermatogenesis. If I ask you on the test, where does spermatogenesis occur? If it were fill in the blank, you could give me a lot of different answers. If it were a fill in the blank question, you could say in the testes. But more specifically, you could say in the seminiferous tubules. But even more specifically, you could say in the germinal epithelium of the seminiferous tubules of the testes. That's where it takes place. Okay. The cells that are in the various stages of development to become the sperm cells are called the spermatogenic cells. Okay. There are other cells in the germinal epithelium besides those that are actually dividing to produce the sperm, and they're called Sertoli or sustentacular cells. I told you there's a lot of new words in here that you probably had never heard of, right? All right. When a male child is born, all of the spermatogenic cells are undifferentiated, kind of like stem cells, but they're, kind of like the stem cells in the bone marrow. They're undifferentiated as far as blood cells are concerned. But then as they divide, they can specialize, right, into the different blood components. Well, it's like that in the seminiferous tubules. All the spermatogenic cells are the same. They're undifferentiated, but when a male is born, and then when he gets to his age of puberty, whatever that might be for that particular person, that's when spermatogenesis starts. And once it starts, it continues daily for the rest of that man's life. Okay, it may diminish a little bit as he gets older, but it's still taking place. Spermatogenesis is still taking place. Whereas in females, the timing of all of it is very, very different than in males. But the divisions are pretty somewhat similar. But anyway, but this is the sequence of events that occur during spermatogenesis. So this is what you need to know mostly about the cell division in the formation of the sperm cells. Those undifferentiated cells are called spermatogonia, and those are the ones that a man is born with. And then as they divide through mitosis, it produces other ones like them. You continue, guys, to produce spermatogonia all the time. New spermatogonia just through mitosis. But some of them that are being produced go on and specialize to become primary spermatocytes. And those are the cells that start meiosis, or the primary spermatocytes. So once they become a primary spermatocyte, it's time to move on through meiosis. Okay? And so they go through the spermatogonia have a total of 46 chromosomes. Because primary spermatocytes are formed through mitosis, they also have 46 chromosomes, right? Okay, and now those are the primary spermatocytes are the ones that go through meiosis. And so as they go through prophase 1, metaphase 1, anaphase 1, telophase 1, that first meiotic division, now the chromosome number is halved, and they become the two resulting cells are the secondary spermatocytes. Okay, and so primary spermatocyte to secondary spermatocytes, they have 23. But what's wrong with them is that they're still in that replicated state. They're not individual chromosomes yet. And so those are the ones that go through the second meiotic division to form a total now of 4 cells that also, they're little spermatids, which is an immature sperm cell. Like it doesn't have a flagellum on it yet and all that. But it genetically, it has everything that a sperm cell is going to have. But they have 23 chromosomes, but the difference is they're not replicated anymore. It's just one strand of DNA, which is the way it needs to be. Okay? And then they'll go on and mature and become motile as they develop a sperm, get all their nourishment. They just mature. They don't divide into this mature sperm, they just mature. Okay? Grow, I guess. All right. And so here's a view of that a little bit. So starting with the primary spermatocyte. So it goes through that first meiotic division to form secondary spermatocytes that then go through the second meiotic division to form the spermatids that then mature into the little sperm cells. Okay. So if you look, I like this picture a lot. What you're seeing is a cross-section of a seminiferous tubule up there that I circled. And see that little orange part of it? They took a plug out of it, and now that's what you see right here. So you're seeing what I've bracketed is that germinal epithelium. And the cells that I'm going to mark in green, and all those. All right, those are all the spermatogenic cells in the varying stages of division to form the sperm. But there are 2 other cells that at first glance you don't even see here. And those are the Sertoli cells, or sustentacular. And they're huge. Here's the nucleus of one of them, and its cell membrane. You see how it kind of wraps around the spermatogenic cells? There it is. So that's one Sertoli cell, and the other one's over on the my left side. So you see there's its nucleus, and then here's its cell membrane. So it wraps around. So those are the support cells. And through hormonal action, they are the ones that actually trigger the spermatogenesis to occur. Okay. So without them, spermatogenesis will not take place. Okay. So they don't just physically support them, they physiologically support them. Okay, and we've already seen this. So the end result after meiosis occurs is that you've got now sperm cells that are genetically different from the original, and they're different from each other. Okay, it gives variety to the species. Okay, so what does a sperm cell look like? Um, these are the smallest cells in humans, much smaller even than a red blood cell. But they're made up of three basic parts: the head, the midpiece, some people call it the neck, and the tail. Um, so you stay on this page. Let's look here. All right. These develop from those little spermatids. So there's the nucleus in the cytoplasm of a spermatid. And as it matures, a lot of the cytoplasm is reabsorbed, and the cell membrane basically just covers this little part right there. So there's very little cytoplasm. The head of it is mostly just a nucleus. Okay. And that head is covered by a cap that's super interesting. This cap's called the acrosome, and it contains enzymes that are critical for fertilization. Y'all have all seen pictures, I'm sure, little videos of fertilization, like under a microscope. It shows the egg, and it's got all these gazillion sperm around it trying to get inside, but only one penetrates it. All right, well, pretend like here's an egg. It's not truly a formed egg yet, but it's an oocyte. But I'll call it an egg for right now. There's the egg. All right, and let's say here's all these little sperm, and just imagine it's millions. All right, well, the egg around it, we're going to see in our next lecture that there's a bunch of cells, other cells around the egg, and there's a little membranous barrier. Well, a sperm has to penetrate all of that, and it can't without the enzyme from the acrosomes of millions of sperm cells. So the enzyme of one sperm is not enough to get through all that barrier. That's why, even though it's only one sperm that fertilizes the egg, if a man's sperm count is low, chances are fertilization will not take place. Even though he still may have a few million, it's not enough because there's not enough of the enzyme from all of the sperm for one to get through. So that's interesting. Oh, and then the rest of it, the mid piece or the neck of it, is just filled with mitochondria, which are very important because a sperm cell requires a lot of energy to move because it has a flagellum. This is the only cell in humans that has a flagellum. It's just like a cilium. The cross section of it is identical to that of a cilium, but it's just longer. And in mammal cells, um, the flagellum is singular. There's not more than one. Like bacterial, there are flagella of bacteria, and there may be more than one. And some protozoans have more than one flagellum. But with humans, it's just one. It's on the sperm cell. But it's used as a propeller, to propel it through. Okay, so enough about the testes and the sperm cells. Now let's look at the rest of the male reproductive organs. Sperm cells, remember I said spermatogenesis occurs from puberty to death. Okay, millions and millions and millions and millions are formed every day. Every day. So they got to go somewhere. And where they go, as soon as they're formed in the seminiferous tubules, they move into a real curled up tube on the surface of the testis called the epididymis. And that's where the sperm cells are stored. They may stay there a few days, and if they don't go anywhere else, then they're just absorbed by the body. Okay. But that's where they mature. That's where they become motile. Is at the epididymis. Okay. So if I have a picture of this? Let me go back. I need to move that sagittal picture down to here. Let's see. Take it down. No, wait. I don't want to do that. Hang on. I will copy it. Copy it? I hope it doesn't copy all that drawing on it. Paste. Yeah, of course it did. All right, maybe I can erase them. Oh, good. Okay. That worked. Yay. I love this smart board. All right, so here's the testis. there's an epididymis, okay, and so that's where the sperm are going to move. And then from there, if emission occurs, then they're going to move up this tube that runs up. That's part of that spermatic cord, um, and it runs up into the pelvic cavity across the top of the bladder and empties into the prostate gland. That tube right there is called the vas deferens, or you can call it the ductus deferens, and it just transfers the sperm to the urethra. Okay, vas deferens or ductus deferens. If a man has a vasectomy, that's named for this organ. And what they do is just go in and snip it right there through the scrotal wall. And so it doesn't mean he can't produce sperm. It's just they can't get to where they need to go. So he's still producing sperm. He still has semen. It just doesn't have sperm in it because semen's produced by these organs over here that we'll look at just right now. Okay. It just doesn't have a sperm component to it. It's just fluid. But anyway, so vas deferens or ductus deferens. All right, and then from there, as the sperm move through, they empty into the prostate gland, and there's a duct that is within the prostate gland. You see it right there. That's called the ejaculatory duct. Okay. Also emptying into that duct is the fluid from this organ called the seminal vesicle. There's actually two of those. They're on the posterior wall of the bladder, but they also empty into the ejaculatory duct of the prostate gland. But the seminal vesicles produce a lot of the components, the fluid components of semen. They have it has fructose in it. Fructose is a sugar. It's a monosaccharide. Remember, glucose, fructose, galactose. And so that gives energy for the mitochondria, or it gives a monosaccharide for the mitochondria to break down to release energy. Prostaglandins are produced there. I've never talked to you about prostaglandins, but they are chemicals that have hormone-like effects in the body. In other words, you have target cells for certain prostaglandins and prostaglandin binds to that, kind of like hormones do. They just are not traveling through the blood like hormones. And the target cells for the prostaglandins from the seminal vesicles are muscle cells, smooth muscle cells, even in the wall of the female reproductive tract. And so once all of that semen makes its way into the female, muscular contractions occur to try to propel the fluid up the female reproductive tract, and that's enhanced by prostaglandins from the seminal vesicle. They also aid in muscle action through the male reproductive tract too to help propel the fluids. And then it's an alkaline fluid. What does that mean if it's alkaline? What's it going to do? It's going to raise the pH. The female reproductive tract, the vagina, is a little bit acidic. Sperm cells can't survive in that. So it's got to have a more alkaline pH than the female reproductive system provides. And so that can be enhanced by this fluid from the seminal vesicle. It's interesting, huh? All right, and then another internal organ is the prostate gland. There's two seminal vesicles, but there's just one prostate gland. And it surrounds the prostatic urethra. The ejaculatory duct is within it. And it produces a thin, milky secretion that is probably the most abundant of all the components, besides sperm cells, of the fluid components. The prostate gland is that. And that helps to nourish the sperm cells. All right, and then another internal organ, that little bitty one right there. There's two of those also, like there are two seminal vesicles. They're called bulbourethral glands. An older name for these are Cowper's glands. But they produce a mucus secretion, and so that aids lubrication during intercourse. But that's also emptied into the membranous urethra. Remember, you got your prostatic urethra, and then there's the membranous urethra, and then from there everything moves through the penile urethra. Okay. So on this next slide, I've laid out for you what the components are for each. The seminal vesicles, it has lots of things in it. And then the prostate is the milky fluid, and then the Cowper's glands release mucus. Okay. And please call it a prostate, not a prostrate. Prostate. That really bothers me. Prostrate is a body position. Prostate is an organ. Okay. Okay. So, semen, the fluid, has various components. Of course, sperm cells, and then the fluids from those three organs. You don't have to know. I mean, we've really pretty much learned a lot of this, the composition and properties, but it's just real interesting. So I left, I've got this in here for you. You don't have to know the numbers that are here. But notice it's slightly alkaline, and that's because of the seminal vesicle secretion, slightly alkaline. It has prostaglandins and nutrients in it. Mucus is in it. And in one ejaculation, and you don't have to know these numbers, but 2 to 5 milliliters. But this is amazing. 120 million sperm per milliliter of semen. Do y'all remember how many red blood cells are in a milliliter, which is a cubic millimeter of blood? Do y'all remember that? It was like 4 to 6 million. Here's 120 million. So this gives you the idea of how tiny these cells are, because we thought red blood cells were small, didn't we? But these are basically the size of a nucleus. Tiny, tiny, tiny cells. Okay. All right. And so this fluid, contains a combination of all the substances from these organs, from the testes, but also the three glands. And it's interesting, the order of release of all of this during emission is, it starts with the bulbourethral gland. Remember, that's mucus. So that's got to be there first for lubrication. Prostate gland, that's the main fluid environment, so that then the sperm will have something to travel through, and it nourishes the sperm. And then the seminal vesicle has all the things that are needed for sperm motility, also for their survival and movement through the female reproductive tract. So I know that's bizarre to know that order, but I want you to know that order. Okay. All right, and so here you see a posterior view. So this is the bladder. And so here I like this view because you can see that there's two seminal vesicles. There's two bulbourethral glands. Okay. Here you can see the testis, the epididymis, and then the vas deferens, and the rest. Okay. Now, if we now look at the external reproductive organs, we've already talked about the scrotum that houses the testes. This is interesting. There is a muscle called the dartus muscle that is periodically contracting and relaxing to pull the scrotum closer to the body wall or farther away from the body wall. So it's basically regulating the temperature in the testes so that it's the perfect temperature for spermatogenesis. This is bizarre, but when I was in high school a real long time ago, that's when guys started, tight briefs got to be real popular. And there were actual articles in magazines about men becoming sterile, men who wore tight briefs all the time. Now, it was temporary sterility, but tell me why. It was too hot, right? It was too hot because the testes were kept too close to the body. And so anyway, but the dartus muscle does that. It regulates that. And then the urethra, of course, passes through the penis. And so the penis has the urethra in it, but it also has erectile tissue. On this picture, you can see the two kinds of erectile tissue. The kind that forms or encloses the penile urethra. So it's this area right here, all this pink. All right, that's called the corpus spongiosum. And then there's two columns on this picture, purple, called the corpus cavernosum. So the corpus spongiosum surrounds the urethra, and the corpora cavernosa are the other two columns of erectile tissue. At the end of the corpus spongiosum, it enlarges, and that's called the glans or the glans penis. And then the skin that overlies that, a lot of times that's removed during circumcision, but that's the prepuce or the foreskin. Um, Erectile tissue is interesting. During an erection, what happens is the arteries leading to that tissue dilate, and so all those arteries engorge or fill up with blood, and that squishes the veins that are next to them, so they basically collapse, and so there's no place for the blood to go, and so that engorgement of blood into the erectile tissue is what produces an erection. I think I've got that in here somewhere. That's a lovely picture. But on this cross section, that would be the corpus spongiosum surrounding. There's a urethra. But those other tubes are blood vessels in the corpus cavernosum. Okay, but this is showing you the cascade of events during an erection, and it's y'all remember way back when we did A&P1, and I talked about sympathetic and parasympathetic responses in with various organ systems, but the one that was left out was the reproductive system. It's because it's a combination of both. Parasympathetic impulses, you don't have to know this for the test, but I just think it's interesting. Parasympathetic impulses are the ones that ultimately cause the erection to take place, but then emission and ejaculation and a lot of the events of orgasm are more sympathetic. So it's both sympathetic and parasympathetic. But basically, in the erection, it's just blood vessels going to the erectile tissue dilate, and the others collapse. So blood fills it. All right. Orgasm itself is accompanied more by sympathetic impulses, and there's a lot of E words. There's erection, emission, ejaculation. A lot of people don't know the difference in emission and ejaculation. I want you to make sure you know these. Orgasm itself is the feelings, the culmination of all of it. But emission is the movement of everything in the man into the urethra, from the organs that produce them, from the epididymis, from the seminal vesicles, cowpers, bulbourethra. All of that movement of the semen into the urethra is emission, and the movement out of the urethra is ejaculation. Okay, and most of that is due to sympathetic impulses. Okay, we all know, whether you're male or female sitting here, you know that females have hormonal cycles, right? Monthly, right? You all know that. Males have hormonal cycles too, but they're daily. And so that's what we're going to finish up today looking at, is the hormone interaction in males. And this is going to take us back to the endocrine chapter and to our lovely pituitary gland. Do you recall that the pituitary gland, in the anterior pituitary, there were several hormones like growth hormone, prolactin, thyroid stimulating hormone, adrenocorticotropic hormone, ACTH. And then there were two others that at that chapter I told you you're going to lump them together and call them gonadotropins. Technically, they were luteinizing hormone and follicle stimulating hormone. Okay, all right, but those two gonadotropins we're going to talk about in this chapter. Okay, one of them in females it's called luteinizing, but in males it's called interstitial cell stimulating hormone, or ICSH. And the other one, the other gonadotropin, is follicle stimulating hormone, whose name tells you what it does in females, but it still goes by that name in males. I don't know why they didn't change the name. But these are those that I referred to back in chapter 13 as gonadotropins. And because they're produced at the anterior pituitary, they are controlled, their release is controlled by a releasing hormone from the hypothalamus. Y'all remember that? Okay. And that releasing hormone from the hypothalamus is called gonadotropin releasing hormone, GnRH, gonadotropin releasing hormone. Okay, so the cycles in males and females starts at the hypothalamus through the release of gonadotropin releasing hormone. And then its target cells are the cells in the anterior pituitary that produce ICSH and FSH. Okay? And those two, the gonadotropins, have target cells in males. They have target cells in the testes. In females, they have target cells in the ovaries. Okay? So today we're going to look at what they do to the testes. Okay. ICSH stimulates the development of the interstitial cells, just like it says. The interstitial cells are also called the cells of Leydig. Remember, those are the ones that are in the testes, but they're not inside the seminiferous tubules. They're in between the seminiferous tubules. And those are the cells that make testosterone. So do you see how testosterone will not be made unless ICSH has gone to the testes? And it won't go there unless the gonadotropin releasing hormone went to the anterior pituitary, right? Okay. So that's how testosterone gets made. So you might wonder, well, if testosterone's made, I thought that was the whole point. All right, but you hadn't made any sperm cells yet. You've just produced testosterone. That's the endocrine secretion. Now we got to have the exocrine secretion, and follicle stimulating hormone is involved in that. As ICSH is being released, so is follicle stimulating hormone, and its target cells are within the germinal epithelium. They are those really weird big cells that surround all the spermatogenic cells, those Sertoli cells. And though it activates those cells so that they will take up testosterone. And once they take up testosterone, then they can trigger the meiosis to begin. Okay? So they trigger spermatogenesis. So it's a combination. Spermatogenesis won't take place in the testes unless ICSH is there, unless FSH is there, and testosterone. See, it takes all of that. It's a little complicated. Probably this is one of the harder parts of the male reproductive system, is just this hormone interaction. Okay? But this occurs daily from puberty till death. Okay? All right. And then the testes, of course, I've already said this, they produce testosterone, right? Which is in the classification, the hormone classification of androgens. This is the main androgen that males produce. Males also produce some androgens at the adrenal cortex, right? But the main place, of course, would be at the testes. And testosterone's roles in a man's body is, of course, the development of the reproductive organs. That's the primary sex characteristics, is just maleness, reproduction of the male organs. But once puberty begins, testosterone also stimulates spermatogenesis, and then also the secondary sex characteristics, which are some of these changes that guys go through at puberty and that are sustained throughout his life, like increased muscle mass, lengthening of the vocal cords, increased hair, facial hair, pubic hair, all of that. That's the male secondary sex characteristics, and testosterone plays a role in that too. Okay. And this, if you know this, that's good. This is the same thing, just much more detailed. Okay, so you could put a big X as far as you don't have to know the detail of this slide on your test. Okay. But primary sex characteristics, you hear about that. Primary sex characteristics, secondary sex. Primary is just the development of the reproductive organs. Okay, and then secondary is all the other changes that males and females go through at puberty. Okay. Now this picture, this takes you back to chapter 13, doesn't it? Remember the interaction between the hypothalamus, the pituitary, and other endocrine organs? But this time is specific for the gonadotropins. So there's the releasing hormone that causes the pituitary to produce the gonadotropins that get into the blood and cause the testes to produce the androgens. All right, and just don't worry about inhibin for right now. We're not going to fool with that. But if there's enough androgens like testosterone, if there's enough of that in the blood, that acts as a negative feedback. It's a negative control, right? So it shuts off the cascade. So that this is why if let's say an athlete's taking illegal androgens, the steroids, they can become sterile. They can also have death results because there's a lot of other side effects. But the reason they can become sterile is because that testosterone or the androgens they're taking, those steroids, yes, they have androgen-like effects in the body, but it's more of the secondary effects, like increase in muscle mass is the big one, blood, all of that. But so they're enough like real testosterone to cause that to happen. But because they're present, the brain, the hypothalamus, thinks that the real thing is there, but the real testosterone can't work. Even if this is identical to it, it can't work because remember, in order to work, you got to have FSH, allowing the Sertoli cells to take it up to cause spermatogenesis. And because you don't have this, spermatogenesis stops. So a man can become sterile when they take those because it messes up the normal cycle. Okay. Next time, we'll cover the female reproductive system.